Bismillahirrahmanirrahim. Assalamu alaikum. It's my second lecture regarding the WBC. In this lecture, we will concentrate mainly the function of the neutrophil and the monocytes. Before going into the details of the function of the neutrophils, I would like to discuss its development also. Regarding the developmental stages of the neutrophils, we have a very important terminology that is called the Ornith count. What is this Ornith count? Actually, it describes the nucleus of a type of white blood cell called the neutrophil in an attempt to detect disease. Again, before discussing this terminology, I am going to tell you that it is not written in Guyton and often it is asked in Viva. And you must know it, that what is Ornith count, because it has a great importance in the detection of the disease process. Actually, it is the developmental stages of the neutrophil depending upon the number of the lobes of its nucleus. I will tell you the details, but in short, Ornith count is the developmental stages of the neutrophil. Depending upon, it is a count which is dependent on the number of lobes of the nucleus of the neutrophil. In general, older neutrophils have more lobes than younger neutrophils. It determines the percentage of neutrophils with 1, 2, 3, 4 and 5 or more lobes. Individuals who have a larger percentage of neutrophils with fewer lobes have a left shift, which can be indicative of disease processes such as infection, malignant tumors, hemolytic crisis, myocardial infarction, acidosis, etc. Don't worry about left shift or right shift. I will tell you all the details of Ornith count, what is that one and what is actually its importance. So, with the time, in the following slides, I will tell you the details. Individuals with a larger percentage of neutrophils with more lobes have a right shift and most commonly have diseases such as vitamin B12 or folate deficiency, chronic uremia, liver disease and so on. Joseph Ornith, because of that we named it Ornith count because of its scientist. He did classify neutrophils into following 5 types. These are the types N1, N2, N3, 4 and 5 depending upon the number of the lobes present. So in stage 1, stages that are the developmental stages. So initially in the first stage the nucleus is unilobed, nucleus is C or U shaped. Normally in normal blood they are 2 to 5%. You can see here the number of lobes of the neutrophil and their percentages. So, normally this type is present just 2 to 5%. If this normal range of these cells it changes, this percentage shows that normally we have this type of the neutrophil the most abundant. But if it is most abundant or it is most abundant or it decreases, that is going to show the disease process. That makes the difference. Again, in other diagram, I am going to show you clearly the number of lobes and development stages of the neutrophil. Principle of the Ornith count is the staging of neutrophil is based on the degree of maturity. The younger neutrophils contain fewer nuclear lobes than the older ones. That's why I'm saying you that it is actually the developmental stages of the neutrophil. The neutrophils enter the bloodstream mostly as bilobed cells, but the number of lobes increases to 5 or more by the end of their short lifespan of 8 to 10 hours. Neutrophils are grouped into different types that are discussed already. The number of granules. The younger cells contain more granules. The size of the cell size decreases as the cell advances on age. This is the graph to show clearly what is meant by shift to left and shift to right. And depending upon the change in the percentage of appearance of these types of the neutrophils. This is the normal count in which you can see clearly that N3 is the most abundant. If it becomes most abundant, that is shift to the left. If it becomes more abundant, shift to the right. Again, what is shift to left or shift to right indicates? Shift to left indicates the total cells counted in N1, N2 and N3 are more than 80%. It indicates that bone marrow is hyperactive. Because left shift occurs due to increased production of cells, this is also called regenerative shift. More immature cells, more early developmental stages cells, they are present in the blood. Conditions related to this type is acute pyogenic infection, tuberculosis, hemorrhage, irradiation. So this is the importance of Ornith count, that it is helpful in detecting the disease process. It different lobes, different developmental stages shows how much the immature cells they are present. Obviously, the immature cells are not able to cope all the functions as the mature ones, or if it decreases or shift to the left, it is an indicative for disease process like acute pyogenic infection and so on. Same for the shift to the right. What it is going to indicate? It is because of the total cells counted in N4 and 5 are more than 20%. The percentage is now changed. They have become more abundant. It indicates that bone marrow is hypoactive. Because right shift occurs due to hypofunction of the bone marrow, this is also called degenerative shift. And the disease may be megaloblastic anemia, aplastic anemia, septicemia and uremia. Now coming to our actual topic, that is the defensive function of the neutrophil and macrophages. Now this word, that is the macrophages, again going to confuse you, but don't confuse. I will tell you what is this one actually. Now we are going to concentrate the following topics. WBC enter the tissue spaces by diapetesis. WBC move through the tissues by embodied motion, and they are attracted to inflamed tissue by chemotaxis. All things confusing, but one by one we will discuss these topics in detail, and also I will show you many animations to clear this topic. Neutrophil and tissue macrophages attack and destroy invading bacteria. First of all, I would like to define what is the terminology, what is these macrophages actually. In the blood, we have different type of the blood cells, including the RBC, WBC, platelets, and so on. Among the WBC, we have lymphocytes and monocytes also. If the monocytes, they remain within the blood vessel, they are called as monocytes. If in case of infection, they come out of the vessel, they gain or they attain water and become larger in size, and now they are called as macrophages. So actually these are the monocytes out of the blood vessel. So at this stage you should not confuse with the macrophages. So I think it is clear now. So the neutrophil and tissue macrophages attack and destroy invading bacteria, viruses and other agents. They can attack and destroy bacteria even in the circulating blood also. These are also suicide bombers, killing invading organism as well as the destroying itself. That is called the extracellular digestion. Monocyte, the largest cell in the blood, having a size of 12 to 14 micrometer. They are produced by the bone marrow from monoblasts. As we have already discussed, they circulate in the bloodstream. They constitute 3 to 8% of the leukocytes in the blood. Now, before discussing the actual function of the neutrophil and macrophages, monocytes and macrophages, I would like to discuss the laminar flow of the blood inside the blood vessel. What is that one? It is written, this whole paragraph that is written in Guyton in the circulatory unit, but what is its importance? That is not written in Guyton. That's I am telling you why this is so. What is the laminar flow of the blood in vessels? That is a specific type of the blood flow flowing in the blood vessels. That one is when blood flows at a steady rate through a long smooth blood vessel, it flows in streamlines with each layer of blood remaining the same distance from the vessel wall. Also, the centralmost portion of the blood stays in the center of the vessel. This type is the flow is called the laminar flow or streamline flow. Laminar means actually in the layers. So, what is its importance? To keep the blood flow in layers, and how these layers they are formed. Actually, we have three layers, two plasma layers outside and one layer that consists of the cellular portion of the blood and remains in the center. So three layers: plasma, central cellular portion, and then again plasma. In simple words, the cellular portion is flowing in the central portion of the blood, while outside that we have plasma all around. And why the nature kept this type of flow? The importance of this flow is that number one thing, to avoid the injury caused by cellular portion to the vessel wall. If the blood cells hit again and again to the vessel wall, they may injure it. So the plasma outside the cellular portion, it is going to provide the cushion to the blood vessels. And the second thing, if in any case these cells come out of the vessel, they have to cross this plasma cushion or plasma barrier. This thing is very important regarding the inflammatory process or the defensive action of the neutrophils, because the neutrophil, they can perform this defensive action while they are flowing inside the blood vessel. But if there is any source of infection out of the blood vessel in the tissue, they have to come outside. This is the thing. This this is the thing. This is the actual topic for today to be discussed in detail. So before that, you must know that neutrophils, they are flowing in the central portion of the blood, inside the blood vessel. And now, if they have to come out, they must come closer to the vessel wall by crossing the plasma portion, and then only then it is possible that they come out. Now coming to the details of all these processes. Macrophages, regarding that one, I told you that monocytes, they are the blood cells which is going to perform the defensive action. They are actually present inside the blood vessel, while in case of infection they come out, they attain excessive water, and their size become larger than the normal monocyte. Now they are called the macrophages. Once they are come out of the vessel, we have two types of the macrophages. Number one thing, these macrophages, they may circulate in the extracellular fluid compartment outside the blood vessel and among different tissues. In the whole of the extracellular compartment, they are moving. But in our body, some of very specific organs, they are the main site for abundant macrophages. For example, liver or spleen or other organs also. So these are the specific tissue macrophages, while the first variety is mobile macrophages, moving all around out of the blood vessel, moving all around the tissues, going here and there. So regarding macrophages, mainly we have two types of the macrophages, either the mobile or tissue macrophages. And tissue macrophages, they are fixed macrophages which are present in the specific organs, specific site, lymph nodes, liver, spleen. All these, they are the major sites constituting the reticuloendothelial system. So these are the major sites where the abundant macrophages, they are present. So they begin life as blood monocytes, immature cell while still in the blood. In tissues, they begin to swell up fivefold, and then they are called macrophages, and they are extremely capable of combating disease agents in the tissue, travel twice the circumference of Earth daily, engulfs and digests cellular debris, necrotic tissue, foreign substances, and so on. And this process is called the phagocytosis. This is the thing. Through this animation, I want to show you that how the blood cells, they are circulating inside the blood vessel. And once they find any infection, they have to come out, and you can see how they are, WBC, they are coming out of the blood vessel into the tissues. RBC and WBC, they are flowing inside the blood vessel, while the WBC, they are coming out of the blood vessel in this way. I will show you more animations. Don't worry, to understand this process very clearly. Actually, what happens, we have the tight junctions here in the endothelial wall of the blood vessel. Whenever there is any infection out of the blood vessel, these tight junctions loosen between the endothelial cells and allow the neutrophils to come out of the blood vessel. Now, at this stage, one more thing should be noticed, that I have already told you that neutrophil and other all blood cells, they are circulating inside the blood vessel in the central portion. Before they come out of the blood vessel, they have to be arranged, or they have to inline to the blood vessel wall. Firstly, from the central portion, they come closer to the vessel wall, and this process is called walling off. After that, these junctions, they loose and allow them to come out. This is the thing. Again and again, I am going to show you how this process occurs. With the diagram, I am going to show you that these are flowing in the middle portion. Firstly, they inline here closer to the blood vessel. Then tight junction, they become loose and allow them to come out. They are coming out of the blood vessel. And what happens at this stage? This is the source of the infection. Here the infective site, it is the infective site. From this source, a special rays spread from this source outside, so that this infective agent acts as sun, and all these the sun rays. These sun rays, they are coming from the main source. You can see it as sun, and in medical terminology we call it chemotactic source. Infective agent, actually at that site, infective site, special kinds of chemotactic substances, they are released. Just like a magnet, this is the magnetic field, and the effect of the magnet, that is spread. It is most abundant closer to the magnet and become less and less as we go away from this source. Why it is so? Because it is going to attract all these cells. Otherwise, what happens? How they come to know where they should go? This is the source. Infective agents, they are abundant here, and at that site special type of the chemotactic substances they are released, and because of that all cells they are attracted to this site. They come to know that this is the infected site. We have to travel towards this site. Firstly, they become in line, and this process is called margination. So, as far as the phagocytosis process is concerned, firstly this process starts and neutrophils, macrophages, all these cells they become active. What happens further? Diabetes, this is the special type of the movement as I have already shown you through this diagram. Through this picture, you can well see this diabetes. They come out of the blood vessel and then they are going towards this one source. So what happens? Neutrophils and monocytes can squeeze through the pores of the blood capillaries. Is this first there is margination when cells come closer to endothelium. So what is margination, how it occurs and why it occurs? This thing, because of that I have discussed this thing previously. To understand the process of margination, what is that when and why it occurs? Because normally they are flowing in the central portion. So number one thing, they have to align themselves along the blood vessel wall. Then what happens? There is some adhesion formation between receptor on cells integrin with selectin and ICM1 of endothelial cells. So they stick with the endothelial cell. There is widening of the capillary pores. Cells move below endothelium and pass through holes in connective tissue and finally out into the tissues. This is another animation to show you the infective. You can see this one as here. Not only the infective agents they are present, but you can I should say you must check the change of the color also here. Redness, more marked area, so that these cells squeeze out of the blood vessel and going towards that site. Some cells move at high velocities. New cell membrane form at leading edge and mid and rear portion of the membrane is absorbed. Pseudopodia attach to the surrounding tissues by receptor proteins present in exocytic vesicles. Secondly, actin in cell polymerize into filamentous network which contracts with myosin. Energy is provided by splitting of ATP. Other cells like fibroblasts, skin cells and embryonic cells also show this movement. So in the end, now they come closer to the infective site to engulf the infective agent. So at this stage we are going to discuss the phagocytosis. It means cellular ingestion of the opening agent, infective agents. Phagocytes are selective of the material depends especially on three selective procedures. First, most natural substances have smooth surface which resist phagocytosis. Secondly, they have protective coat that repels phagocytosis. Obviously, they are not offering the phagocytosis process. They will resist. But how they resist? Through which things they are going to resist? So they have mostly number one thing, most natural substances are smooth surface. which resist phagocytosis. Normally, why this process not occur? Because they have the smooth surface. So, if any agent or any infective site, any tissue have irregular surface, it is indication for the injury. Secondly, they have the protective coat that repels phagocytosis. Normally what happens? We have the protective coat. If due to the disease process, due to the infective agents, this protective coat is damaged, obviously they are offering the process of phagocytosis. Third important thing is Ig antibodies attach to the bacterial membrane on one side and to C3b on other, which attaches to receptor on phagocyte membrane. This process is called opsonization. If you don't if you cannot, if you are not able to understand this third point, don't worry. This process is discussed in the next chapter in much more detail. What is this process and how it occurs? If it is not clear to you at this stage, don't worry, we will come to this topic again. Through this animation, I want to show the process of the phagocytosis. You can see the bacteria or infective agent. These are the cells, defensive cells, and like that they are engulfing. They are firstly they are coming to the infective site, infective agent, and after they come in contact with that one, they engulf, they digest. Now the next thing is how the phagocytosis occur through neutrophil and macrophages. Through the neutrophils, number one thing is attachment and recognition. And for recognition, I have already told you that if there is a smooth surface, it is not attractive. So, if there is not smooth surface, if their protective coat is damaged, all these things, because of that, they are going to be, they are going to recognize the infective site, and after that, they will attach to that one. Neutrophil first attaches to the particle by surface receptors. For the engulfment, pseudopodia project out via membrane remodeling, cytoskeletal changes, and polymerization of the actin. Pseudopodia meet and fuse over it. It's better to show you in the animation. Then killing and lysosomal bactericidal enzyme, they are secreted and like that. You can see this one. Neutrophils, they are coming on contact with it and they engulf, and the remaining part, that is pus. Pus is actually the dead infective agents. You can experience small pustules over your face also, and if you puncture this pustule, the pus is relieved. And what is actually that one? That is the end product of the infective agents once it is attacked by our defensive defensive cells. For the macrophages, they are immature cell but more powerful, can engulf larger particle. That is the thing must be noted here. Again, lysosome and other granules fuse with the phagosome and for digestive enzymes, and then digestive vesicle is formed. Macrophages can extrude the residual products also. This is almost the complete story about the inflammation. Firstly, margination, then they come out to the infective site, engulf phagocytosis occur, kill them, and the end product, that is pus. At this stage, I am telling you the first line of defense. Before that, I must say that this is a very simple process about the inflammation. This is the first stage how the very simple inflammatory process that is dealt with our defensive functions, defensive cells. But it is the first stage where it is the initial and very simple process, but it is not the end of the story. The process of defense starts from here, from the neutrophil and these macrophages, and go and become more and more complex if the infective agent is more powerful, high in number, more stronger. So whole story changes. So it is the just beginning of the defensive story, not the end. The end will occur after the end of the next topics following lectures, when we will discuss the role of antigen antibody reaction, complement system, and all these complex defensive system of the body. Up till now, I have discussed the initiation of a very simple or the simplest process of inflammation. But it is not the whole story. I will tell you as we proceed how you can number the line of the defenses. So here I must tell you the initiation of the defensive process. It occurs through the mobile and the tissue macrophages and the neutrophils. Very simple, but up till here. It will be changed. It will be much more complex with the complexity of the infective agents. Damage, extent of the damage occurring to the body tissues. So this defensive process is just not so simple. It changes depending upon many things, including the number of the infective agent, extent of the damage, how much the damage occur, how much the stronger, and how much the complexity of the infective agent, maybe bacteria, viral, parasite, complex, combined infection, then all whole of this defensive story changes. Accordingly, the main target is just to defend, to save our body from the infective agents. But this story changes. Our line of action, that will be changed with the complexity of this infective process. Okay, for that, keep with me and keep watching my lectures up to the next lecture. Thanks and Allah Hafiz.